question being presented in this research study is, what is the origin of causation for structurally defective centrioles found in tumors? And the experimenters formulated a specific hypothesis, and because it is known that the overelongation of centrioles could be associated with mitotic polo-like kinase, or PLK1, which is responsible for the regulation of centriole elongation in human cells, the experimenters predicted that abnormal PLK1 levels could be responsible for the defective centrioles produced in these tumors. So the experiment that researchers designed was to observe the effects of various lab-induced disruptions to different phases of mitosis that were specifically targeted as phases in which elongation typically occurred. And to look at how different disruptions that were produced in the lab to these different phases of mitosis affected the process of centriole elongation. In order to do this, they first obviously identified where in the cell cycle elongation most commonly occurred using EM analysis. And then they utilized MLMC-D3 cells and RPE1 cells to actually visualize how these defects impact ciliation. And to do all of this, they first created a cell culture and synchronized the cells using a specific drug treatment and mitotic shakeoff. And then they utilized immunostaining and immunolabeling in order to actually visualize centriole elongation at different phases in mitosis. And they used wide field microscopy and structural illumination microscopy to observe the actual centriole elongation in a given moment, and then followed by storm imaging and EM analysis. And then the centrioles were measured, and the resulting data and imaging produced from all of these methods was statistically analyzed for significance.
What is the origin of causation for the structurally defective centrioles found in tumors caused the researchers to turn their attention towards PLK1, which has shown to associate with the centriole elongation factor, CPAP. What researchers ended up finding out was that PLK1 plays a dual role in the centriole cycle. Researchers were also able to find out that PLK1 overexpression and inhibition can influence procentriole elongation. Researchers were also able to provide an explanation and proof of why overactive PLK1 is frequently associated with centriole structural abnormalities. The researchers were also able to provide a demonstration from their data of why numerical and structural centriole abnormalities often coincide with tumors. What ended up happening and new information found out was that PLK1 has previously been shown to promote the biochemical maturation of centrioles. PLK1 is also able to stimulate elongation of procentrioles, while at the same time promotes their gradual maturation, which ultimately in the end reduces their ability to elongate. What researchers ended up also finding out was that PLK1 could represent an effective way to couple these two processes and asserting some consistency in centriole size during cell cycles.
The interpretation for this article is that abnormal centriolar structures are associated with cancer. When mitosis happens, any defects in centriole structure can result in genetic abnormalities during cell division, which may lead to tumor development, meaning that this person may have a higher chance of getting cancer. Therefore, by preventing such defects in centriolar structures, the chance of getting cancer can be reduced, and one such target could be the PLK1, which can therefore be used in cancer therapeutics to reduce the genetic abnormalities that cause cancer. The prolonged G2 phase of the cell cycle also results in the elongation of procentrioles. Any chemical or stress that delays or impacts the cell cycle will have a negative effect on the centriole structure. The scientists tried to understand this by identifying the mechanism, which is that the elongated procentrioles retain proximal end functionality, but distal ends could not form cilia. This essentially means that the proteins could not bind together, leading to their impaired functionality. The PLK1 and 2 play a role in the elongation of the centrioles. Inhibiting the PLK1 inhibits centriole over-elongation, likely via interaction with CPAP. Multiple regulators play a role in elongation of MT, as PLK1 inhibition alone did not completely prevent centriole assembly and elongation, because cell cycle-specific regulators affect the centriole structural defects. Numerical and structural abnormalities of procentrioles are interlinked, and the variability of length of the centrioles is considered normal, and therefore the cell has a high tolerance for a variability in size, but exceeding that normality will lead to improper chromosome segregation, leading to tumors. Mother centrioles can also get matured, be stable, and not easily be impacted by the changes in the cell cycle, but the procentrioles are comparatively more sensitive to these changes.
So finally, I did the conclusion. And basically the question we're asking is, what biological conclusions can we draw from this experiment? First of all, human cycling cells don't have a set structure for monitoring central elongation, which means that there's no one mechanism that ensures that centrioles are formed at an exact length, meaning that there are many factors involved. This causes the elongation process of centrioles to vary throughout the cell, which is what the researchers were primarily studying. Researchers predicted that these varying elongation patterns could signify that elongation factors vary in concentration in different locations within the cell. Researchers were able to speculate that any event that causes mitotic delay may potentially promote centriole over-elongation, providing a cause for this phenomenon. Below, I have a quote from the actual study, which states, centrosome numerical errors can originate from aborted mitosis, from cell fusion, or from centriole amplification. Our data imply that all of these events can promote the formation of centriole structural aberrations. This can explain the origin of aberrantly structured centrioles in tumors and why centriolar structural defects are frequently found in tumor cells, along with numerical defects. And basically, to conclude, this study and its results explain the origin of centriole variability in cells and why that and other centriole abnormalities are associated with overactive PLK1. This demonstrates why centriole abnormalities are often found in tumors.
I researched what is next, so these are some things that came to my mind after reading the article, or some things mentioned in the article by researchers that were still unclear, and these are things they could do to continue this study. One thing is to determine if PLK1 hyperactivity has any other effects on centrioles other than just over-elongation and altering the centriole structure. What other cell structures could PLK1 hyperactivity have effects on other than centrioles? How PLK1 actually regulates procentriole elongation, examining exactly why these centrioles lack a mechanism that monitors elongation, and kind of going along with that point, determining a means of incorporating an elongation monitoring mechanism into these centrioles in a lab setting of some sort, and then studying these effects of having such a mechanism. What does this do to help the centrioles? Because this over-elongation is a result of the lack of an elongation monitoring mechanism. So having this mechanism, how would this impact the centriole as a whole, since it's not able to over-elongate? Discovering the extent to which cells can tolerate variations in centriole length without harmful consequences. So basically, how long can these centrioles get without it having a negative impact on the cell as a whole? Whether and to what extent PLK1's functions in procentriole assembly overlap with those of other polokinases. And then finally, determining why the percentage of cells that form over-elongated and structurally inconsistent procentrioles, why does this vary among cell lines? So these are just a few ideas that researchers could use to continue this study.